But it's an honor to really introduce this panel here. So moderated by Dr. Karl Pfleger, joined by Dr. Matt Kaeberlein, Dr. Kristen Fortney, and Dr. Barzilai. So please take it away.
Okay, so this is a super important panel because clinical endpoints are extremely important, so that's why everyone should come in from the break area. And I am super honored to be moderating such an amazingly esteemed panel. We're gonna start out a little, basic and backgroundy for anybody in the room who's not intimately familiar with the field, and then get into some really important stuff. But let's do quick intros. Each of you just introduce yourselves very briefly, and let's start with Matt.
Sure. Matt Kaeberlein, CEO of OptiSpan. Is that brief enough?
Amazing.
Kristen Fortney, CEO and co-founder of BioAge.
Near Barzilai, the president of the Academy for Health and Lifespan Research.
Wow, that was faster than I expected. Okay, so we're going to start out with a little bit of basic background. So Kristen, can you just explain for, not the aging field, but just in general, how endpoints work in normal clinical trials? How are they chosen, and sort of how do we measure clinical success?
Yeah, for sure. And to be specific too, we're going to talk about FDA approvable endpoints, right? Like what suffices for a drug to be approved by FDA. And really they're looking for a few different criteria. One is that it has to be meaningful to the patient. For example, improve survival, improve function, improve how they feel. But equally importantly, it has to be validated and well understood. So they are looking for a kind of scientific consensus. There's a marker, and they are looking for something that if you measure it over and over again, it's not going to vary too much. And finally, what they're really looking for is that it be related to the disease, so they understand how it's related to the disease. So these endpoints, they do come in different flavors, right? Like for heart disease, for example, they're looking at a composite of mortality and whether you get strokes, and that's actually really hard. You need big trials to see that because that's fairly noisy to measure. But then on the other end, you have things like an obesity where it's just your weight on a scale, or even diabetes where it's a blood biomarker, HbA1c. But it's a blood biomarker where they really understand how it's related to the disease itself, to blood glucose, and where it's really been shown that it predicts, basically how you progress and your risk of other, worse things happening to you.
And is there anything interesting that you want to add from looking at it from that was sort of from the FDA's perspective, but I think you covered most of the important stuff, but is there anything interesting to add in terms of how the companies normally think about how they want to choose the endpoints?
Well, from the company's perspective, you're looking to do it as efficiently as possible, right? So you have limited money in the bank, and you're like, well, how can I get conviction around that my target is doing what I think it's doing in the clinic as rapidly as possible? So usually by the point you get to a phase 3 trial, you're going to FDA, you're investing a lot in your program. But from a company perspective, you want to go and do a human experiment as efficiently as you can to convince yourself that your drug is doing what you think it's doing. So this might be something completely different, right? It might just be a very simple blood biomarker or something functional that would not suffice as the regulatory approvable endpoint, but gives you confidence to continue to invest in your program.
Okay, so now we're going to move into actual aging. So Matt, can you just give us the basic background on what is the geroscience paradigm or geroscience hypothesis?
Sure. I don't like hypothesis because I think it's been proven, but
I'm using paradigm now after your paper from a while ago.
So I mean, I think first of all, I would just like to add one little bit of context, and correct me if you disagree, but I think we tend to think about endpoints in the context of disease, but it doesn't have to be a disease endpoint. So for example, you could, as Restore Bio attempted to do, show you can boost vaccine response. It's my understanding wrinkles is even an endpoint for FDA, so it doesn't have to be that disease connection. And I think as we're thinking about geroscience applications, we may want to think about clinical trials for endpoints outside of specifically the big killers. Okay, so the geroscience concept, let me put it that way, really is pretty straightforward. I think we all agree that biological age is the single greatest risk factor for a bunch of bad stuff. 9 of the top 10 killers in the United States, a majority of functional declines and diseases that are relevant in developed countries. And geroscience is the biology that connects the biology of aging to those declines in functional outcome and disease. And if we can be successful at understanding that biology and targeting that biology with interventions, we can slow the onset and progression of all of these functional declines and diseases simultaneously, which is great because that gives us a whole plethora of endpoints and indications that we could go after if we have successful geroscience interventions.
Great. And okay, Nir, so very high level, doesn't have to get into a lot of details, but does the field have evidence in support of this idea, and sort of what does that vaguely look like at the high level?
Yeah, sure, but let me just start with something, some observation here, and picking on something that was said already. You three are PhD, and we're talking about clinical indications. And I want to make this point. How many here are MDs? Raise your hands. Okay, not a majority here, but when we go to medical school, the first day they teach us do no harm, okay? That means that on a Gauss, half of the people, you'll come with headache, they're not going to give you Tylenol, because Tylenol, 1 of 100,000, will cause a liver problem, right? We are conservative. The second day of medical school, they say there's no always and there's no never in medicine. And so not only you're conservative, you're confused. And I'm saying that because your PhD don't have this. You're saying, yeah, we cured mice, let's go to humans, right?
We learned confused in the first day in PhD.
But the point is different. It's not about you. The point is different. The people at the FDA went only to the first 2 days of medical school. Okay, now it's wrong. Of course, it's wrong. And Zan is sitting here, and Zan knows more about everything than we do. Okay, but I just want to say that when we're talking about these things and we're going to the FDA, the people in the FDA are conservatives and they don't believe that we're safe. Okay, pretty much. The nice thing about the geroscience hypothesis came up with the hallmarks of aging, okay? And the hallmarks of aging are basically something that goes wrong with aging, and if we fix it, our animals or humans are increasing health span and increasing lifespan. The nice thing is that the drugs that we are using, and we are pretty sure they're targeting the aging, are targeting the hallmarks of aging. Met have published the hallmarks of aging with rapamycin, right? It almost touches every one of those hallmarks. We've published about metformin, it touches the hallmarks. It's true to GLP-1. It's true to a list of other drugs, even if there's no evidence for that. So I think there's a total link between the gerence hypothesis, the hallmarks that we think are important. By the way, the hallmarks are not the mechanism. They're not all the mechanism, but they're important to target. And the fact that drugs are touching the hallmarks and have effects on multimorbidities and overall mortality.
And can you talk a little about your work summarizing the efficacy of, with respect to these concerns of existing drugs?
Well, for those
Very
of you who haven't been here yesterday, I showed the evidence on clinical trials on 4 drugs that are, changing the hallmarks of aging, increasing lifespan in animals, and in clinical trying have effect on multimorbidities and overall mortality. And those included SGLT2 inhibitors, metformin, a GLP-1 agonist, and biphosphonates. And rapamycin didn't make on the scale of 12, didn't make it because there's really not enough clinical trials, certainly on the doses that we want on rapamycin, but there are 10 other drugs that are potentially really targeting the hallmarks of aging and are
Great.
relevant.
Kristen, endpoints and trials for new aging interventions, do they look similar to those in the non-aging space? And do companies that are in our sector plan to use new drugs against multiple age-related diseases?
Yeah, sure. So, I mean, right now there's no path for FDA to approve a drug for aging. If there were to be in the near term, it would probably be a very complicated metric where you'd need a lot of people. Like if it's going to be a biomarker or a composite mortality endpoint, these are very large trials, right? So those of us in biotech, because these, per the geroscience hypothesis, a lot of these targets that are relevant to aging are also relevant to several specific diseases. And it's usually most efficient to choose that initial disease and go there first, where you can run a very small controlled trial, not break the bank, get some early, and of course, even though it'll be for a particular disease with a particular disease endpoint, you can also measure your aging biomarkers along the way. And this is historically what's happened, too, with multi-indication drugs. Like, there's many multi-indication drugs. That idea itself is not new. The favorite example of our field is the statins, right, which were first approved for a fairly rare disease, familial hypercholesterolemia, and are now used to prevent disease in most older people. And we don't really like that example because it took 20 years. Like, it takes a very long time to go from that very small population to a very large preventative one. So I think that's what we have to think about. How can we really accelerate that timeline? But there's a lot of examples that are contemporary as well, like the GLP drugs, for example, the weight loss drugs in diabetes and obesity. Generally, you have to run additional phase 3 trials. That's typically how it goes. Sometimes you can borrow from the trials you've already done as part of that evidence package. Like, I think with SGLT2, that was important with some of the renal approvals. But basically, we're trying to pick a particular disease, ideally a large one to get, so that you don't have to start from too small a population and expand from there.
Nir, great, that was great, and I think that's the sort of paradigm we have now. Nir, can you explain to everybody, because I'm sure there's a few who in the audience who probably don't know, the basics of the TAME trial, what the idea was, and sort of how the endpoints are structured?
Sure. The TAME trial was designed for FDA approval as aging is an indication. Okay, I want to make sure that you understand that. The trial was designed because it was done already and we knew that it's going to be successful. The question is, how do we have a template for any pharmacy to do the trial? And it was a breakthrough, I should say, in our thought process, because we had to be agnostic for the disease. We don't know what disease you're going to get next. If your mother was diabetic and you're obese, you're going to get diabetes. But we have a drug that whatever disease you're going to get, we're going to delay it. So we clustered those diseases together. The advantage of clustering disease together has to do with what you said, the expense of the study. If you take people over the age of 65 and you're looking at several diseases, you need less people, okay? If you have one disease, you need more. On the other hand, we had to cluster them in a way that no single disease will break through. Let's say we start TAME trial with metformin, and in a year, you have 30% significant decrease in heart disease. You'll have to kind of stop the study, and you wouldn't. So the statistical calculations here were really very severe and thoughtful. I would say that Lilly, for example, is interested. Now Lilly has everything, and they kind of understand that they have a gerotherapeutics. They want to do a TAME trial. Actually, they wanted to call it a TAME trial, okay? Using pretty much our indication and show a thing. So I want to say another thing. The FDA, so we came to the FDA. There's a beautiful movie of Ron Howard, The Edge of Aging. And you can see how we went to the FDA and how they interviewed the people in the FDA. And the FDA said, yeah, that's great, bring it on. At the end, the FDA, at that time, we said that's how we target aging, and they said, no, don't say aging. If you have a drug that prevents a lot of diseases, that's okay. And we discovered that we can move on. We can move on with everything. We can call it aging, and whatever the FDA wants to call it. It's like pornography. We'll know what's going on.
And the time frame is still 7 years? Something like that?
Look, the power of the TAME trial was we should have achieved those goals within 4 years. We asked for a 6 years funding just because we didn't know how it's going to go.
Right, okay. All right, great. So essentially we have a model. It still takes a number of years, but in some ways it's better than the multiple phase 3s. Matt, can you discuss Loyal's success with the FDA in their dog therapeutic program, and then also whether or not you think that success in the dog aging project will lead to a change in the way that drugs can get approved for aging in pets?
Sure. Yeah, and so just to maybe start with a high-level picture, I think companion animals, so dogs, cats, pets living with their owners, are a very powerful approach to FDA approval. If you don't know this already, FDA regulates medicines in dogs and cats and other companion animals very similarly to how they're regulated in humans. You go through an approval process, you get FDA approval. I think because companion animals age 7 to 10 times faster than people do, there are opportunities to do true, certainly longevity clinical trials, which would be hard to do in a healthy population in humans in a reasonable time frame, and get statistical power. And so that was the idea behind the Dog Aging Project rapamycin clinical trial. We could do a study in a few hundred dogs in a 3-year treatment period, or 1-year treatment, 2-year follow-up, and have statistical power to see changes in lifespan as the primary endpoint, and then health span metrics as secondary endpoints. So it's a very powerful system in which to do that. Loyal is a company out of San Francisco that is moving forward with a couple of drugs that they are moving into the FDA conditional approval pipeline. I think they're likely to get conditional approval for one of their drugs this year or next year. That would be the first approval for lifespan in any animal, including humans, obviously. But people may not realize this. We may already have our first geroscience FDA approval. A company called Trivium Vet out of Ireland has gotten approval for rapamycin for, in this case, hypertrophic cardiomyopathy in cats. But they started with rapamycin through a geroscience perspective, looking at multiple endpoints. They settled on hypertrophic cardiomyopathy as their first endpoint, but they're looking at other endpoints. So this has sort of gone under the radar, but that may actually be the first geroscience FDA approval to have actually been successful. Again, it's conditional approval. I didn't say what that is. And that's something that I think we should think about in humans, but it's better established in the companion animals, at least at this point, where you can actually get approval based on safety and reasonable expectation of efficacy without doing the phase 3 clinical trial. That gives you, I think, 5 years on market to, and you have that time to do the phase 3 clinical trial. So Trivium's product is already on market or will be soon. If Loyal gets conditional approval, their product can go to market, and they've got 5 years to do the efficacy trial to prove efficacy. So that's a unique sort of pathway to get the approval in place, and then you have the time and resources to move forward and demonstrate efficacy. We may want to talk about this later. There are obviously some potential risks to the field. If a drug is approved as a longevity drug and then turns out not to show efficacy, I think we should be thoughtful about what that means, but this is the way FDA is operating today. And then I'll just come back to the Dog Aging Project. So there have been all sorts of challenges.
Matt, can you just say what's the drug?
Loyal's drug?
Yeah.
Yeah, so it's called LOY-001, and it is an IGF-1 antagonist. So they're trying to reduce IGF-1 signaling. They've got another LOY-002, which is a metabolic activator.
And it's specifically for the big dogs.
Yeah. But my point is more in the other session they asked about centenarians in women. So women centenarians who have the lowest IGF-1 levels, they're already centenarians, okay? They're already 100 years old. They live twice as long if they have the lowest IGF-1 level. 60% of the longevity genes that we find, actually I should say differently, 60% of female centenarians have some antagonistic to IGF growth hormone actions. And the point here is when you have evidence from genetics, okay, the pharmaceuticals, FDA, everybody wants this evidence from genetics, and 2/3 of the drugs were approved to FDA, not for dogs, but have approved, are based on genetics finding also. So I think this story goes, and we use, when we discovered that, we used IGF receptor antibody that Amgen had in mice, and we showed that we elongated lifespan by 10% and improved health span significantly. So this is good news for the field.
Yeah, I think that's really important because I think the study that you just mentioned that you were involved with, the genetics and the fact that big dogs have more IGF-1 signaling and age faster than small dogs all contributed to this reasonable expectation of efficacy in support of the conditional approval.
So it was conditional approval for lifespan.
To be clear, that hasn't happened yet, as far as I know, but they're, I think, getting close to
Yeah,
that.
the tweets, I think, suggested that that piece was done and they were just waiting on
So it's
GMP or something,
it's
but
a
I don't
little,
know the details.
yeah, it's a little bit unclear, and again,
okay.
my understanding, and I could be wrong, is that the tweets have been talking about actually the second drug, and FDA has approved the clinical trial design. So when people say that the FDA has approved the trial, that just means FDA said, yeah, you can do the trial. But I don't think, and again, if anybody knows different, please correct me, but I don't think they've gotten the conditional approval yet.
But to your point, the Trivium one is a great example where they do have conditional approval, and I think that would be really meaningful, because as you were mentioning, there's just not enough data on rapamycin yet to give a lot of people conviction. But if this hypertrophic cardiomyopathy trial reads positive in a large population of animal pets, that could really change, I think, how pharma thinks about it too. Do you know when that's reading out, Matt?
The pivotal?
Yeah.
I don't even know if they've really started it yet. Yeah, because they just got the conditional approval a couple months ago.
All right, so in some ways, lifespan as a readout is like just admitting that mortality is bad, but we can, that's already
Mortality
an endpoint
is bad,
That's
yeah.
already an endpoint that's okay,
It
but it
checks
just
the
takes a long
box.
time.
It's like unambiguous, right? Like the you can all agree that like mortality is meaningful. Yeah.
Okay. All right, so of course the problem with mortality is that you have to wait a long, especially in humans, is you have to wait a long time. So, so I'm going to open this question up to everybody, but I want to just sort of point out that Matt is one of the people who's talked about the lack of the problems with the existing biomarker, the clocks. But we have a number of different biomarkers that we could potentially use, that the field wants to use eventually as substitutes for lifespan and mortality, notably the clocks, but there's a whole bunch of others. And but they maybe aren't ready for prime time. So when do each of you think that the field will have some type of age-related biomarker that could be a substitute as a clinical trial endpoint? What kind of timeframes do you think we're looking at before that's actually viable?
Can I start? Because
Yeah,
I often get mischaracterized as to what I actually say. And I say a lot. Okay, I get it. But so it's not that I don't believe the clocks don't work or that they're not useful. I don't believe that the direct-to-consumer, the rush to market with these things is helpful, and it's being done in a very sloppy way that I think is actually damaging the ability to get these things accepted and validated so that we can actually use them in clinical trials. I think the clocks are extremely powerful. I don't know that they work well enough yet to, because I think this is all about really, and again, you guys may know more about this than I do, but at the level of the regulatory bodies, it's about convincing them that they actually work, right? That they actually are sufficient predictors of quality or quantity of life for people. And I think that they're not to the point yet where you can go to the regulatory bodies, put a body of evidence together that these things have been validated to that level. My concern is that all of the noise and nonsense that's happening in the direct-to-consumer space is actually going to make it harder to convince the serious people at FDA that we're serious people and that these things actually work. So I don't know what the time frame is. I would hope within the next 5 years, for sure, we should be able to make that case. I mean, I think the and I'm not talking specifically about epigenetic clocks, but when we look at the biomarker space in general, I think we have some really powerful tools, including the epigenetics, that are very predictive for health outcomes. The other thing that I'll just say, and this is my pet peeve, we should just stop calling it biological aging. Like, you can't have 150 different tests that measure 150 different things, and they all are measuring biological aging. These are predictors of health outcomes or mortality. And I think if we frame it that way, we sound more rigorous and more serious.
The clocks are super interesting, carry a lot of information. I guess I don't think they're going to be part of an FDA approval process in the next 5, maybe even 10 years for a number of reasons. They don't check all the boxes that FDA wants to see. I mean, first of all, there's no scientific consensus. There are conferences every year, and everybody has their own favorite biomarker because they all carry information, right? So it's hard to get everybody aligned around one, but that's one ingredient we would need. Another is that they are often highly variable, with different people, different days. And there's also no clear relation to the disease. Like, FDA cares a lot about this, whether they should or not is a separate question, but that's something they definitely care about. And these are often sort of more black boxes that carry a lot of information, but we don't completely understand why, So that's one thing. So I think these are still really useful to measure in the clinic. I think they can tell you a lot about your therapy and what it's doing. but the path to an approvable endpoint is very hard. So that's sort of one thing. But I also want to point out that I think that that's okay. That I think there's like other biomarkers that will get at what we need, or other things we can measure like along the path to something more like measuring aging. And to be very concrete, right? I mean, there's endpoints like NEARS, which is really looking at a composite of disease. FDA does that all the time, right? So that's one path you can certainly do. Maybe not as your first indication, but if you're going back after your first approval and want to do a large trial to get that aging designation, like that's a real route. And I also want to point out that like along the path from where we are today to something more like aging the indication, there are goalposts like frailty and sarcopenia, right? There are things that are not aging itself, but are not the diseases we have today, that are a bit more tractable, a bit more concrete, where you can measure things like physical function or muscle mass, right? And these aren't aging itself, but they're sort of like along the way. And that's where I think, I hope that there is an approvable path for a sarcopenia drug and a frailty drug in the next 5 to 10 years. And I do see concrete progress there. Yeah.
And this is from, and we'll get to you in a minute, Near, but this is from a woman whose amazing company in some ways started from clocks, so she knows, and who uses it for therapeutic development, even if there's
Yeah, I don't want to
skepticism
dump on
timeframes.
clocks like I love clocks. That's how we get conviction around which mechanisms to bet on, how to evaluate them in animals, and how to get early signals in the clinic, but regulatory approvals are different.
All right,
Yeah.
Near.
Can I just ask Kristen to expand on one thing she said, which was the connection to the disease? So do you mean because we don't have a clear mechanistic direct, connect the dots? So for example, if one of the epigenetic marks was regulating a gene expression that was clearly involved in the disease, that would potentially set the stage more so? Is that what you're saying there?
That would help a lot, right? Because HbA1c in diabetes, that reflects your blood sugar levels of the last 3 months. It's like they really understand mechanistically what it means. Yeah.
So some of like the Vadim Gladyshev lab stuff on which of the CPTs are causal might. Right.
Yeah, that's right. It's not like that it's impossible. There is a path there through better understanding of what the biomarkers are doing.
Near.
So I'm one of the guys that measured 10 biomarkers, and I'm anywhere between 20 years younger than my age and 4 years older than my age, right? So that gives you the whole really. I would agree with that. You are. Thank you. If I average that, it's not so bad. But look, what we need to do is not only have biomarkers for aging, but also biomarkers that change with the intervention that we want. And that's very important. It's important for the consumer, right, to see that something is like the cholesterol, right? It's moving. It's important for the biotechs because they want in phase 2 study to actually have it move to a different direction. And that's why we started an initiative that's called FAST. It's at the end of a FAR now, but it might be moving on to ARPA. God help the government and ARPA. But this is the idea. There's a DPP trial with Biobank that looked at metformin lifestyle intervention and placebo, ended in 4 years ahead of time. Plasma and blood are stored. There is a lot of SGLT2 studies. There's a lot of GLP-1 studies. And we're going to take the blood, the plasma of the first year intervention and see not only what the biomarkers of aging, but what have changed in a relatively small time. And I think this would be really very important for the field. Each one of the drugs might do their own things, but there'll be a biomarkers of aging that will be probably common to all of them. So that'll be 5 years, I agree, okay? That'll be 5 years. I was saying that on TAME, so don't so don't hold me for that, but I think it's within the reach of 10 years, or 5 years. I just want to make one point. When we use hemoglobin A1C or cholesterol as a biomarker, then every lab in the world has to measure it the same way. And that's what the FDA will approve. So having biomarkers, I think, will allow us some confidence, and we can use whatever biomarker we want, but it's still not FDA approved. The FDA didn't want biomarkers for TAME. We told them we're going to find biomarkers for TAME, but they didn't want it. So the clinical things are still important.
Okay, so this is sort of the last big question. And I want to, ideally, any of you who has something to say on this. So if you could change the current regulatory system to whatever you wanted it to be, we had a version of this question of one speaker yesterday, but let's see what you guys have to say. You know, what would you change? What would you, if you had a wish list or you were king, what would you change about the current regulatory system?
So I want to say the following. Instead of saying what, I want to say that FDA at the top levels are ready to do a lot for us. It's really the lower level, the bureaucrats that are making our life impossible some ways. So I just want to say this.
So she'd be like Trump and get rid of all the bureaucrats.
I wasn't being political here, but I want to say something that you didn't ask, but it's very relevant. We didn't talk about nutraceuticals, and nutraceuticals are a problem, and I think they maybe cause more harm than good. But there are nutraceuticals that have been studied in clinical studies, and I think it's important that the FDA will recognize, or there will be a way to come with nutraceuticals that have shown an effect. And it will push also more companies to check those nutraceuticals. As an example, Mars, the chocolate bar company, have had 20,000 people in a study where half of them got the flavonoids, the chocolate powder, and half of them got a very tasty chowder that didn't have. And in a 3.5-year study, I think Cosmos, they showed endpoints, okay? Now, Mars cannot go on a candy bar and say this is prevent cardiovascular disease and mortality, but this was important. There are lots of NMN companies that are actually going on kind of an FDA route, but if we could have the nutraceutical part in the FDA as a new ID, that would be great.
So I think this question, to really answer it, would require a lot of smart people and a long time. But there are a couple of thoughts.
We have 3 minutes.
Yeah, I know. I'll keep it short. So we already heard, I think, yesterday about 2 proposals that are sort of being floated, one from A4LI, one from Kaeberlein. I think they both have important components to them that I support, and so I think encourage everyone to check those out. And then I think it was Randy who said during the panel discussion this morning, really changing the incentive structures is, I think, what we really need to change. And there are all sorts of incentive structures that favor incremental advances, very, very expensive clinical trials for people with very severe disease that increase, their lives in a poor state of health by a couple of months. And so that's at the regulatory level, it's at the big pharma level. That's where I would really focus my attention first, is how do we incentivize geroscience therapeutics to improve healthy lifespan? What do we need to change in the incentive structures to do that?
Yeah, so I agree with everything that's been said by Nir and Matt, but just to add a couple things, like if I could wave a wand today, I would love there to be a path forward to get a drug approved for frailty or sarcopenia, because this is very related to incentives, right? If there's a path, then people are willing to invest to see if the drug will go there. Without a clear path, that puts off investors, that puts off pharma, no one invests in the space. And I think there really are a lot of mechanisms in our field that could have efficacy there if only there were a path. And that should be doable in the near term. And the other thing, which I think is happening to some extent, gets to, you've got your drug approved for one indication, now you want to expand to a whole bunch more. How do you do that as rapidly as possible? And there's concrete proposals on the table like Nir's, but there's also now starting to be pressure from some of the big players because we have drugs like that in hand, right? With the GLP-1s from Lilly, with SGLT2, these pharma companies are running these huge trials and they're having these discussions now. So I think there's reasons to hope that there could be a more efficient path to get that multi-indication label more efficiently in the future, which we really need.
I want to make one comment. We discussed it in private before, but I think frailty, sarcopenia, those things are not good for us. They're endpoints. It's hard to revive those muscles. It's hard to revive the frail patients. I think we should really think more of, we don't want to treat sarcopenia, we just don't want to have that. And I think to focus more on prevention when we design studies is really important, because we've been failing in sarcopenia. Although we think we have that, we've been failing. And frailty is even more complex.
There have been no trials there.
Well, there was no phase 2 that went on, right?
There was no phase 2 tried, right? So it's not failures so much as like lack of experiments that were tried. Yeah.
The timer says 18 seconds. Do we
I
have
just
time
had one,
for a-
one thought, because
All
I
right,
think in terms of going back to incentives, once we get these things approved, I think we also need to be thoughtful about how do we incentivize making this available to a large swath of the population. I think the GLPs are a really interesting example where, at least for a while, they were pretty cheap, and now the questions, because of compounding, they were allowed to be made by compounding pharmacies, and now there are questions around, will that continue? So some thought needs to be put there, because we will be successful, guys, and when we are, we want this to be available to a lot of people, not just a small minority.
Dylan, do we have time to open it up to the audience? One question. All right. Irina got shut down earlier, so.
Kristen, I empathize with your jet lag. That's the first thing. Then just to have a quick comment, the reason that clocks cannot be FDA approved is because they cannot be tested in blinded randomized studies. You need to tell how old you are, so then it could be predicted with variable, not very high accuracy. So that's probably the major hurdle. And they overlap predictions for healthy people and patients who are of the same chronological age. So then my main thing is 2 years. Imagine that there are people who, and Kristen, imagine that there are people who have sarcopenia now are at the threshold of developing it. Prevention probably is not going to work for them. Why can't we use that as one of the major outcomes?
Not prevention, but
Not prevention, but like for people who are at the cusp.
yeah. So who don't sort of progress to the next phase, right?
Don't we already have biomarkers
Yeah.
such as sarcopenia reduction and systemic protein changes related to sarcopenia?
Yeah, no, I think that's right, and there's precedence for this in a lot of other diseases too, right? You sort of divide it up into severity, and if you take a more severe subset with events more likely to happen, you can have better power. So, and I think that's a good first way to approach it. Yeah.
One more? Nope.
No mas.